Welcome to day 2. We're going to start off this morning with a series of very short talks. The first session will be a series of demos, and our first talk will be Irene Papatheodorou.
Thank you. So first, I would like to thank the organizers for dedicating a session on the portals. I'm Irene Papatheodorou, and I'm from the European Bioinformatics Institute, and I lead the gene expression team there. I'll mainly talk about the EBI's single-cell expression atlas. But first, I'd like to, just for 1 minute, to talk about the portals of the Human Cell Atlas. So we are a whole community of bioinformaticians and software engineers, and we meet regularly. You can, through this URL, at the data portal of the Human Cell Atlas, you can actually see all the different portals that are involved. And here we're going to do short demos of 4 of them. So first, I'll start talking about the work in my team. So since 2009 at the EBI, we've been developing the Expression Atlas, which is EBI's resource for transcriptomics and proteomics data, where we reanalyze them and we try to show in an intuitive way gene and protein expression in different tissues, diseases, and conditions. The use cases we try to fulfill is questions such as where is my favorite gene expressed and how its expression changes in a disease, for example. And we traditionally deal with bulk RNA-seq, microarrays, proteomics. At the moment, we have over 3,500 studies from 62 different species. And so the way the data flows into Expression Atlas is like this. So we collect datasets from archives that are of high quality. We perform systematic analysis, and then we show the abundance and localization of genes and proteins. So that's a bit of background. So very recently, so last year, we expanded the Expression Atlas to deal with single-cell RNA-seq data, and we released a new service within this resource, the Single-Cell Expression Atlas. You can see it's a URL down here for a direct link. So the Single-Cell Expression Atlas helps users discover and interpret gene expression analysis results at the single-cell level. At present, it holds 123 studies from 12 different species, including the main model organisms, such as mouse, worm, Arabidopsis, and fly. We have close to 1 million cells, and we do have, at the moment in production, 7 Human Cell Atlas datasets from the DCP, which you can access here at the bottom of the page. We also have the Malaria Cell Atlas and Tabula Miris. So zooming in to the front page, so this is the main query where you can search for your gene of interest and see where it's expressed. Here I selected a gene called REG1B that you put through and hit search. So what you get initially is a results page that lists the experiments where this gene is expressed. So you can see the species. Here it's only human, but you could find the same gene in different species, whether it is a marker gene in some cluster, what's the title of the experiment, and what are the variables of the experiment and the number of cells. And so these top 2 experiments, REG1B is actually a marker for some of their clusters, or one of their clusters. And then on the left, you can filter this list by the cell type that you're interested, or the tissue, or just check and see only the experiments where it's a marker gene. So if we now just go into one experiment, I select this one. It's an HCA experiment of the pancreatic, of the human pancreas. When we go inside the experiment page, we basically see the same TSNE plot side by side. On one side, you can color the cells according to the clusters or the metadata. Here, I've colored them according to the cell type. And also on the other side here, you can perform a little gene query. So because you search already for reg1b, you see with blue the cells where reg1b is expressed. And the darker the blue, the higher the expression. So you can see here that it's concentrated really the expression in this cluster that then we find out that this cluster of cells is labeled as pancreatic acinar cells. So on this side here, you can also see a heat map with the marker genes, and also you have various options to look at the experimental design, download more files of the different types of analysis, and some supplementary information with links to the source where we got this data set, whether it's RExpress, GEO, or the DCP, and so on. And yeah, that's an idea of what the heat map looks like of the different clusters. And we show by default the 5 top genes that come up from our analysis with ScanPy that has been run in the background. And you can also download the full table, and there are various other ways to do that. So finally, I would like to, so that was a very quick demo of the website. There are lots more data sets to discover in there, and I would encourage you to have a look. Here, we have also developed workflows where we retrieve the data from the Human Cell Atlas DCP, and we perform our analysis, or other data production workflows that we generally use in the Single Cell Expression Atlas for data sets that come from other species or other sources. And we have actually put those tools and workflows within Galaxy so that anyone can use them and run them, or even combine them. You can combine, for example, tools from Surat and ScanPy and run them through the Human Cell Atlas, use galaxy.eu. And finally, I would want to close with some future work, where we're going. So we would like, we are working towards developing a special view for the HCA data in collaboration with the Sanger Institute in Kingston. This is what we have nicknamed the Kingston portal so far, and will be released in early next year. And also in the meantime, we're working towards developing these more detailed anatomograms that are interactive and zoom down to the single cell resolution. Here are just 2 examples. So we will be releasing some of this through our website, but also freely for other users to embed in websites with links to the different cell types when these are known. Finally, I would like to thank my team. Those 2 people in red are here, so Matt and John, so you can find them in the breaks and discuss. And also our funders, especially Wellcome Trust, as this was Single Cell Atlas is funded by the Wellcome Trust. And our collaborators, and Alvis Bresma as well, who is also here, so that's how he's in red, and our collaborators from the Sanger. Also, please, you could follow us on Twitter to find out about releases and courses and training that we provide. And yeah, thank you very much.
We have time for 1 or 2 questions.
Yeah.
Is this only for Human Cell Atlas data, or can also external people submit their data to this repository?
So we retrieve data from other repositories, so such as GEO, RExpress, and the DCP as well. So we, if a data set is of high quality and it's in a GEO, and it's not an HCA data set, and it's in GEO or RExpress, we will probably find it and process it. So we've had members of the community, especially from the plants community, and Fly come to us and ask us to include certain data sets. But yeah, at the moment we cover 12 species, but really we could cover any species that are in ensemble. So yeah, we can do anything. Yeah.
You said you're including high-quality data sets?
Yeah.
Do you have like strong exclusion-inclusion criteria or thresholds?
So we have, we do have some threshold criteria. So we do QC and data sets where the cells are of poor quality. We exclude them.
But you would exclude data sets, like single-cell data sets with a few number of cells or too few sequencing depth, or it's more like the quality control on the cells.
Yeah, so we exclude data sets that are very few cells, or the low quality of the reads, that we don't get good alignments and so on.
Thank you.
Let's thank you again.
Thank you.